In this video, we are going to talk about colony morphology. And basically what that means is we are going to talk about different ways that we can describe and classify colonies that grow on an agar plate. So if you'll recall from the last video, we did streaking for isolation. And that was really important for several reasons. One is so that we can start to identify bacterial species, because if we have a mixed sample and we want to know what's in them, we can streak that sample onto an agar plate, incubate the plate, and then look at all the colonies that grew, because most organisms grow uniquely on an agar plate. So the trained eye can sometimes just look at a colony and say, ooh, that's E. coli, or ooh, that's Staphylococcus aureus. Now, sometimes it's not that simple, but once you have an isolated colony, you can then run a bunch of biochemical tests to figure out what organism that is, or if it's a newly discovered organism, figure out how it works and which other organisms it's related to. So some of the things we're going to be looking at is the shape of the colony, color, size, elevation, all sorts of things. So let's go ahead and look at some of those categories. So listed on this slide are 7 kind of different characteristics or categories that people typically think about when describing colony morphology or how a colony looks. Now, in a virtual environment, things can be a little bit more complicated because you can't manipulate the agar plate to really look closely from different angles at a colony. So I will make sure that on any online assessment, I give you an image that is very clear, okay? Harder to misinterpret. But we'll talk a little bit more about that on the next slide. So size is one way that we can characterize a colony, and it's normally just on the diameter of the colony. And some people, will have a specific scale and say this is exactly, this many millimeters or centimeters. And then we can say, it's small, medium, large. If it's really, really tiny, we say it's punctiform or pinpoint. But it can be, at least as far as I know, there aren't clear cutoffs on, this size to this size is small. And maybe there is. In my personal line of work, I studied one organism, so it didn't really matter how big it was compared to others. So you won't need to know, any specific measurements that indicate a size. But just know we do look at that, especially when we're comparing different colonies on a plate. What we do use, in my opinion, more often is shape. And so with these, you want to make sure that you don't get too over analytical because it is easy to make things more complicated than it should be. So we're going to start with this top row here. And these are some common shapes. And the most common, at least in this course, is the round colony. Another term we use is circular. And before I go on to the others, I want to point out the text over here. Do you see how there are bold words and then sometimes there are unbold words? You're always going to want to use the bold terms. Those are the micro terms, okay? So if you look at this particular shape right here, pretend you're looking at a colony and you're like, oh yeah, okay, it's this one. If you wrote down spherical, you're not going to get any points for that. That's not the term we use, okay? You would have to say round or circular to get credit for it. Now, you're never going to see a spindle, so you can kind of ignore that one. It is common in the micro world, just not on the ones we'll talk about. I'm going to come back to irregular. Let's look at filamentous. So this one reminds me of like spokes on a bicycle. If you'll notice, they all these little lines meet in the middle, okay? And each of these radiate outwards, okay? They don't They might bump into each other, but they don't really overlap. They don't branch off from one another. Okay? It's just they start in the middle and each little line radiates outward. That's filamentous. Rhizoid is more like tree branches or a tree root system. Because notice, again, it all merges in the middle. But if you follow one out, check out where my mouse is. If you follow one out, notice that this one, it branches off here, it branches off there, 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 there. Right? So it's more like either the roots of a tree or you could say the branches of a tree. Irregular, we're going to use this term pretty loosely. If the colony isn't round or filamentous or rhizoid, we're going to go with irregular. And that just means it doesn't have a, kind of like a normal shape. So that's going to be irregular. Now, some people do classify really tiny circles as punctiform. Notice that's the word I used for size as well. And when I was trained personally, we only use punctiform for size because you'll notice next to it, common terms are tiny and pinpoint. That's a size, right? But in some books, they do lump it in to be like a tiny circle. So if you put punctiform for the shape, I'm not going to count off, assuming it is a very tiny circle. And you will see some pictures of what punctiform versus round looks like. But just so you know, even if I were to show you a picture of a tiny round colony, if you just said round, that would be fine. Okay? All right. So that is just the overall shape of a colony. And there will be plenty of pictures coming up to help you see some examples. The next category is margin. Now, with margin, you are only looking at the edge or the border of the colony, okay? You're not paying attention to what the entire shape is. You're only zooming in to see what's happening at that edge or border, okay? So if it has a nice, smooth, continuous border, notice the term is smooth. Also entire. Again, those are interchangeable. So there's no irregularities, right? It is just a straight line. It's curved, but it's straight, smooth, uninterrupted. Okay? I'm going to kind of, again, skip over real quick to the terms we've heard before. Notice what's happening with filamentous here. Again, we're only focused on the edge of the colony. Do you see how they stick out? Again, they radiate from the middle. This one looks kind of like a sun. Okay, so again, straight lines projecting outwards, filamentous. If it's branched, right, so they're radiating outward, but they're branched, that's rhizoid. Again, notice only the edge of the colony looks like that. Now, the next two, people tend to get a little confused. So lobate here is where there are lobes. If you kind of follow like my mouse is, like let's go right up here. See how it's a big round one, big round lobe, okay? That's gonna be lobate. There are clear lobes on the edge of this colony. Irregular is when it's just kind of all over the place, okay? There's not a bunch of clear big lobes. It's not filamentous, it's not rhizoid, it's not smooth. What are you left with? Irregular. Now, I do want to kind of mention real quick with the lobes, notice that they're pretty thick, especially if you compare them to the margins of the other options. These lobes are pretty thick, okay? Notice that it almost looks like they're like fingers, right? Kind of pointing off the edge. That's going to be the big difference between lobate and irregular. Now, not shown on here, let me see if I can get this pen to work. Not shown is undulate, which is wavy. So for this one, let me see if I can get it on my first try here. It's going to be much smaller and thinner than the lobes. Pretend those aren't pointy. Okay, pretend they're nice and like round. Okay, that would be undulate. Okay, so they're much more narrow. There's a lot more of them compared to lobate. Okay, see how with lobate, we've got a big old lobe here, another lobe there, another one here, bigger one there, okay? So that's going to be the difference between undulate and lobate. And y'all, I'm not trying to trick you guys with these. If I were to show you a picture of a colony that is lobate, I'm not going to include undulate as an option, okay? Because the goal isn't to trick you. I just want you to know, like have an idea of what lobate margins look like. And you need to know that lobate is a term you only use when talking about the margin of a colony. So in my opinion, probably the hardest part of Lab Exam 1 is keeping all these terms straight. And I don't just mean what does a round colony look like, but knowing which term goes with which category. So if you look here at entire, this term is only used when categorizing the margin. So on an assessment, I may show you a colony and say, what is the shape of this colony? And I might put entire as an answer choice. Well, you can rule that out immediately because entire is not an option to use when describing the shape of a colony, okay? So that's where you're gonna have to read the questions carefully and say, okay, she's asking for shape. My only options are round, circular, irregular, yada, yada, yada. Okay, entire is not an option when describing shape. All right, let us continue. Let me get this pen off of here so it's not distracting. Okay, so surface and texture, these are much more subjective categories in my opinion, and the scientist in me doesn't like subjective. But take a look at them. So for surface, smooth, rough, wrinkled, another term for wrinkled is rugose, shiny or dull. So honestly, it's just like when you look at the surface of, a penny, you're like, ooh, that's shiny, or ooh, that's dull. The same thing. You're just, when you look at the surface of that colony, how would you describe it? Does it look wrinkly? Does it look smooth? Okay. Now for texture, think of if you were going to touch that colony. In lab, we never touch a colony, but if you were going to, how do you expect it to feel? Okay, so moist is going to be the word you would use if you think it looks like it'd be kind of wet. Again, wet is a common term. You don't ever use common terms, especially on your unknowns. You always use micro terms. If it looks like it'd be sticky, then the term you write is mucoid. If it looks kind of buttery, the term you use is butyrous. It doesn't look dry. Now, sometimes words will kind of overlap, right? Because maybe the texture is smooth. All right, well, that's fine. I'm not going to count off if you choose a subjective word that happens to fit in both surface and texture. Just be careful when you do that, okay? All right, elevation. So for elevation, this is where you're looking at how the colony grows up off of the plate. So if you are in lab, I would tell you to hold the plate up at eye level and describe how the colony is formed. The vast majority of the colonies are convex. They're like a little dome. Okay, so right here. They come up and off the auger like a semicircle, like a dome. We call that convex. The other type we tend to see in lab is umbonate, where they look like a fried egg. Sometimes you'll see flat or plateau, where it kind of starts off curved and then it straightens out, okay? You very rarely see these ones on the bottom, okay? So when it comes to your unknown number 1, which you'll be getting soon, these aren't ones you're going to need to worry about, okay? They exist, but they're not ones that I'm going to put on your assessment. You really want to focus on these, okay? Now, number 7, and I'm sorry the captions are covering that. I meant to scoot this text up. Another useful category is color. However, most colonies are considered beige, or if you want to use the term off-white, that's fine. But the point is they really don't have a color, okay? They don't have a pigment. So Normally, you're going to say your color. I usually just say beige. Now, there are a few, including some of the unknown organisms that you'll see, that do have a pigment. They grow a gold colony. There's one that's a blue-green color. There's another that is, well, you don't have this one for your unknown, but there are organisms that are a red color. There's one that's bright yellow. So when you're thinking about color, try not to overanalyze it. If it's obviously blue, then put blue. But if you look at it and you're like, that's some shade of white, tan, beige, that's fine. Just say beige and move on with life, okay? There's no need to like really worry about that because the point is that there's no pigment. Now, you don't want to use translucent and opaque if you're describing the color, because there is no color translucent. There is no color opaque. Those terms are optical properties, right? You can see through the object, or you can't, but they are not colors, right? So especially when you get to your unknown report and it says what color is your colony, please do not write translucent and do not write opaque. All right, so let's take a look at some pictures here. So for this one, this is really driving home how diverse colonies can be in size. And I won't be asking you to do this on your unknown report or on any assessments because I could be showing you organism A, which is punctiform. But if I zoomed in on it, right, and put my camera right up close to it, I could take a picture that makes it look small or medium or even large, okay? So I won't ask you to do that, but I just want you to see that this scale on picture A is the same as the scale on picture B, C, and D. So you can see that some colonies are really tiny, some are really large. So we can use size to classify them. All right, so here's what you're gonna have to be able to do on the exam and on your unknowns. Now I give you the answers right off the bat, but that's okay. I do recommend when you're studying to cover those up and see what you get on your own. Now, the key to success is to always look at an isolated colony when you're trying to describe it. Because if you look, oops, where's my pen? Hold on, sorry, technology is not cooperating. If you were to look, sorry, y'all. Click, there we go. If I asked you to describe the colony morphology and you looked right here, right, which is really 1, 2, 3, like 4 colonies merged together, that is very different than, let's say, this colony right here, which is all by itself, okay? So you always look at an isolated colony. So let me get rid of these head marks. So I would actually recommend looking at this left-hand colony because notice this one has 2 colonies pretty close by. So I circled that one for a reason because I want you to notice point number 2, don't overanalyze. So we do classify this colony as round. You can actually see another isolated colony right here, right? If you look at that, that looks pretty darn close to a circle. But if you're overanalyzing and you're looking at this colony right here, you're going to be like, what are these little horns that it has, right? Nope, it's just another colony that's kind of close. So again, the benefit of always looking at a single colony, but if it's not a perfect circle, that's okay. We still call it round. It'd have to be like a really wacky shape for us to call it irregular. All right, so now I asked about the margin. So remember, we're only looking at the edge here, okay? Do you notice that the edge is a nice, smooth, straight line, right? I'm gonna kind of trace it, right? It's a nice, continuous, straight line, so it's entire. To me, the surface looks smooth. The texture, it kind of looks like if I touched it, it would be tacky. So I would call that mucoid. And again, you may choose different words as long as you're on the right track. If you told me that the surface was wrinkly, then you'd lose points, okay? But if you chose something other than smooth, depending on what it was, probably okay. Again, don't overanalyze it. Now, for elevation, this is where you want to kind of look at how it's growing up off the auger. Easier for this one in person. But if you kind of put your pin here on the top of this colony where it looks like it's the tallest, and you kind of sketch a line down to where it's at, you can tell that this does make a nice little dome, right? So it's a nice semicircle, so that would be considered convex. Okay? All right, so we'll do another one. So if you look at this one's shape is round because if you trace the outside of it, and I'm not going to be very good tracing it with my mouse, but right, it's round. Okay, pretty darn close. So it would be round. However, when we're looking at the margin, just the edge, notice it radiates out, straight out. Now they might touch each other, that's fine, but that is going to be the definition of filamentous. Remember, rhizoid, which is the one people tend to confuse with filamentous, is where it branches off from a given line, okay? So this one doesn't, it just kind of looks like it's hairy, and so that's going to be filamentous. Now, I would say that this looks smooth surface to me. To me, it looks like it might be dry. Again, it's a picture, it's a lot harder to tell, so just give me your best thought. And then for elevation, this one is hard. Again, limitation of a picture. So I would accept personally 2 answers. And the reason it's hard is because this middle area, let me get my mouse with it, this middle area right here, if that is just like a reflection of light or something, then it's possible that this is flat, right? It's just bloop, like a pancake. So I would accept flat. However, if it's lighter in the middle because it's actually a little bit raised, right? And if it's only raised in the middle, then it might be, If it's only raised in the middle, then it would be umbonate because this part on the edge is very clearly flat. So if it's raised in the middle like a fried egg, then you could say umbonate. All right, so last slide, I just want to show you guys how diverse colonies are. Okay, all 4 of these pictures are organisms that belong to the Bacillus genus. Okay, and so notice just how different. These 4 organisms are closely related to each other, but they grow so differently on an agar plate. And so this is why colony morphology is really, really important because knowing what they look like can help us identify what we're working with in a sample. All right, y'all, that's it for this video. Please let me know if you have any questions.